So hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Students, I hope all of you are doing great and amazing. Right? This is a very important chapter. So all of you students, if I'm audible to all of you, and if the presentation behind me is visible to all of you, quickly add any emoji in the chat, students. Add any emoji in the chat if I'm audible and visible to all of you. Now, students, this is a very important chapter because this chapter will completely change the way you understand biology. You will learn something called as what is haploid and what is going to be diploid. So if you really want to understand what is mitosis, what is meiosis, and what exactly is cell division, this is a particular session for you students. So good evening all of you students, good evening all of you students. Hello. How is everyone doing students? How is everyone doing? Before we start, one second students. Students, I hope I'm audible to all of you. Students, I hope I'm audible to all of you. Yes or no? Perfect, students, perfect. Now, students, we'll start the class with a very simple activity. Very simple activity. I want all of you to name 5 different animals for me, students. Or plants also works. 5 different animals or 5 different plants for me, students. In the chat, all of you. I want all of you to name 5 different animals or 5 different plants or a mixture of those two. Right? Now, let's say lion, right? Mango plant. Let's say a bacteria, let's say any type of protista, let's say dinoflagellate, right? Goniolix. Let's say one of the fungi, right? Any of the basidomyces fungi, right? So when you name all of these organisms, students, every single organism, all the way from a small ant to a large elephant, every single animal is actually in the starting made up of a single cell. Yes, even me, even you, all of us were a single cell. For example, in our case, it is zygote. But all of us were made up of a single cell. Now, from a single cell, one tiny cell, how did we, students? From a tiny single cell, how did we become, right? One single tiny cell, that is your zygote. How did we become 3.4 trillion cells? Trillion cells, students. Trillion cells. How is it possible, students? How is it possible? From one single cell to 3.4 trillion cells? Well, the answer is very simple. The secret behind that entire, rapid multiplication of cell is called as cell division, students. It is called as what? Cell division. So that right there is the secret behind the entire process. Now, students, how many of you love playing games? Raise your hand, students. How many of you love playing games, students? How many of you love playing games? All of you? Good evening, students. Good evening. Hello. Okay, students, if you love playing games, right? Obviously, you would have fallen somewhere. Let's say you play cricket, you play football. Every single time you fall down. And every single time you fall down, you end up getting a small wound on your hand. But eventually, that wound will heal up. Now, what is the secret behind that, students? What exactly is the secret behind that? The secret behind that and this is actually cell division. That is, one single cell will replicate, it will divide and become 2 cells. That is, from one single cell, it will divide into 2 daughter cells. 2 daughter cells. That is called as cell division, students. That is called as cell division. It's a very easy chapter, but before we start, I want all of you to understand a few terminologies here and there. Okay, students, how many of you know the meaning of haploid? Raise your hands. Students, how many of you know the meaning of haploid? Students, quickly in the chat. How many of you know the meaning of haploid? Very simple, students. Haploid basically means a cell or an organism which has a single set of chromosomes. Yes or no? Right? Cell or organism with a single set of chromosome. Now, what do you mean by that? For example, in the case of humans, students, we are diploid. It means we have 2n. So we have 23 pair, 20. So we have 23 chromosomes, 23 chromosomes. So in total, we have 46 chromosomes that is diploid. Half of it is haploid. That is n basically means 23. Now, diploid means 46. Then what is triploid, students? What do I mean by triploid? Anyone in the chat? Triploid. Now, diploid basically means cell or organism with two set of chromosomes. I am not talking about two chromosomes. I am talking about two sets of chromosomes. One set is 23. Second set is 23, 23, 23, 23. Right? So, two set of chromosomes. Right? When I say two set of chromosomes, I mean 2n is equal to 46. That is double the number. That is the double the number. Now that's the meaning of 2n. Now students, what is going to be 3n? Can anyone tell me in the chat the number of chromosomes, right? The number of chromosomes which we can see in triploid. Anyone? Triploid. How many number of chromosomes? Just again double it and tell me in the chat, students. Double it and tell me in the chat, all of you. Now, what exactly is chromatin? Students, chromatin, chromatid, both are two separate things. Centromere, centrosome are two separate things. So please listen to me very, very carefully right now. Right? What is chromatin? Chromatin is a thread-like structure. It is a thread-like structure which is made up of DNA plus histone and non-histone protein. Non-histone protein. Now, students, I'll give you a better perspective. Students, I'll give you a better perspective itself. What exactly is chromatin? Okay, just give me a minute, students. Just give me a minute. You'll understand better what exactly is a chromatin, students. Okay. Now, students, I want all of you to focus very carefully right now. All of you know, students, this right here is our DNA. Right. This right here is our DNA. This is the DNA molecule. All of you know it has nitrogenous bases. It is made up of nucleotides. Nucleotides, the backbone is sugar phosphate. All of you know that. Nitrogenous bases are here, students. Now, if I take this DNA molecule, long DNA molecule is there. Now, students, if I wrap it around a protein here, okay. Now, this is called as histone. Now, if I take the DNA here and I wrap it around it, okay. Now, this is your histone. Individually is called as histone here, histone, okay. Now, when I wrap it around it, this entire structure is called as nucleosome right now. It is called as nucleosome. So, long DNA, histone protein around it. Now, this entire blob, DNA plus histone is called as nucleosome right now. Now, students, if I make a long thread of nucleosome, can all of you see, students? Long thread of nucleosome right now. Long thread of nucleosome. This right here is called as chromatin. Now, this right here is called as chromatin. Chromatin is a long thread-like structure which is made up of nucleosomes, DNA plus histone and non-histone proteins. Okay. Now, students, now let's take, I want all of you to understand, if this is a chromatin right now, this is the thread here. This is a thread right now. This is the chromatin thread. Okay, this is a chromatin thread. Now, if I take the chromatin thread and I completely condense it, what do I get? Can anyone tell me, students, in the chat? Can anyone tell me, students, if I take a chromatin thread and if I completely condense it, what do I get? Like I take a thread, nicely condense it. For example, students, let's try to give it a shape right now. Let's try to give it a shape. This is a long thread and I will start to coil it around like this. Okay. The thread is coiling right now. It is getting condensed. The thread is getting condensed right now, students. Students, what do I get here? Students, what does it look like right now? Tell me quickly in the chat, students. Quickly in the chat. If I take the thread and if I completely condense it, this is what I get. So from one chromatin, the thread, long thread, this is the chromatin. This chromatin has the DNA in it. DNA plus histone and non-histone protein. Non-histone protein. Okay. Now if I take this chromatin and if I give it a shape, if I condense it, it will give you a chromosome. So all of you know right now what exactly is a chromosome? All of you. So chromosome is nothing but it is a structure. It is a structure condensation after chromatin. So if I take a chromatin, if I condense it, I get a chromosome. Now the question arises, do you find chromosome all the time? Students, do you find chromosome all the time? No, not really. You cannot find chromosome all the time. This structure called as chromosome, it is only visible. It is only visible during the cell division. When the cell is dividing, it is undergoing prophase, metaphase, anaphase, telophase. That is when you can see the shape. Otherwise, in normal condition, this is how the DNA is going to be. In normal condition, this is how the DNA looks like. It looks like a chromatin. Okay. So I hope all of you, this much information is clear to all of you. If it is clear to all of you students, right? If it is clear to all of you, now let's go ahead right now. Now I want all of you to understand this particular structure is called as what, students? It is called as chromosome. All of you know what is a chromosome right now. Okay. Now students, in this chromosome right now, in the middle, right in the middle, there is a primary construction. Right in the middle, there is a primary construction here. Primary construction. Construction means made up of heterochromatin. It is made of heterochromatin in the middle. Okay. Primary construction called as centromere. So this right here is centromere. This is called as a chromosome. Right in the middle is called as centromere. Okay, centromere students. Now listen to me very carefully. Now I want all of you to focus on these two structures here. These structures here. These two structures are not called as chromosome. Individually, they are called as chromatid. They are called as what? Chromatid. So these are called as chromatid. This is called as a chromatin, and this is called as a centromere, and this entire structure is called as a chromosome. Students, how many of you understood till now, students? Raise your hands. All of you understood? There is a very basic difference between chromosome, the entire structure. Chromatin is the long thread. This is right in the middle, we have centromere, and these two individuals, right? These two individual structures here. This is called as one chromatid, and this is called as one more chromatid. One more chromatid, students. One more chromatid. Now a question arises, students. A question arises. Sir, then where are genes? What are genes? Where are genes present? Students, if you take a small segment here, if you take a small section here, somewhere here, we can find the genes also. We can find the genes also here, somewhere in the middle, students. Okay, so somewhere here you will find the genes also here. Genes are actually present on the DNA. Small segment of a DNA is called as a gene. Okay, small segment of DNA is called as a gene. Now students, let's get to the cell cycle, the chapter name. Right, a cell cycle and cell division are two separate things. First of all, I want all of you to clear your mind and tell me, cell cycle and cell division, two separate things. Cell division is actually part of cell cycle. Cell division is actually part of cell cycle, students, part of cell cycle. Now, how do I understand, students? Now, a typical cell cycle has three parts. A typical cell cycle, students, has three parts. The first part is called as cell growth. Cell growth. Second part is called as DNA replication. DNA replication. And the third part is called as cell division. This is where the cell divides. So cell division is actually a part of cell cycle. So cell cycle has how many parts, students? A cell cycle has three parts. The first part is called as cell growth, where the cell actually is growing. When I say growth, I mean increase in cytoplasm. Increase in the cytoplasm. So there is an increase in cytoplasm here. There is a increase in the cytoplasm happening here in cell growth. Now DNA replication, what do you mean by DNA replication? All of you learned about DNA replication here and there, right? The increase in DNA content. Let's say students, here I have a chromosome like this. See students, all of you please understand, this is a chromosome, this also is a chromosome. Both of them are chromosomes. Both of them are one single chromosome itself. Now a question arises, sir, how can I identify if something is a chromosome? If you want to identify if something is a chromosome, look at the centromere number. Okay students, now all of you tell me, how many centromere do I have here? All of you in the students, all of you in the chat, tell me, how many centromere do I have here? I have one centrome here. Right in the middle, one centrome here. Now students, here also have only one centromere also, one centromere. So in both the cases, understand students, in both the cases I have only one centromere, one centromere. So it means that in both the cases I have a single chromosome itself. Now then what is the difference then? Here also have one chromosome, here also one chromosome. The only difference is that number of chromatids. Okay, now if you go back here students, if you go back here, I told you these two are called as chromatids. Now students, if two chromatids, if they are part of one centromere, two chromatids part of one centromere, like one family, they are also called as sister chromatids. Then sir, then what is non-sister? Then students, what is non-sister then? Very simple. So this, understand. The meaning of non-sister is very simple. Let's say I have one chromosome here. I have one more chromosome here. Okay. Now there are two different chromosomes. Now this chromatid and this chromatid, one from this centromere, one from this centromere. Now these are called as non-sister. They are called as non-sister chromatids. They are called as what? Non-sister chromatids. So is that clear to all of you students? I'll upload today itself, students. Okay, I'll upload today itself, students. These are called as non-sister chromatids. Basic difference is clear to all of you. Okay. Now students, if you come back here, in DNA replication, what is happening? In DNA replication, the number of chromosomes will remain the same. In DNA replication, students, please listen to me very carefully, very important. Okay, in entire DNA replication, number of chromosome will remain same. Number of chromosome will always remain the same. So if the number of chromosome is always the same, then what is the changing part here? The changing part here is the DNA content. See students, all of you tell me in the chat right now. Okay, now if you go back to this image, right now, okay, now if this half is not there, okay, let me just draw this diagram one more time for you students. This diagram I'll draw one more time for all of you students. Tell me which one has more DNA content. Tell me students, which one has more DNA content? Okay, DNA is, chromatin is present here, okay. Now this one has a single centromere here, one chromosome. Okay, now let's draw one more chromosome here, and this time this is the DNA content here. This time students, tell me in the chat which one has more DNA content. Tell me students. If I am switching from here to here, tell me students. If I am, this is one chromosome, This is also one chromosome. Both are one chromosome itself. Both are one chromosome itself, students. Both are one chromosome itself. But here DNA content is comparatively less because here we only have one chromatid. Students, let me make it bigger itself. You're like, sir, you have drawn it very tiny. Okay. Now in this, if you look at this one chromosome because of single chromatid, because of single centromere, because of single centromere itself. But students, you tell me in the chat right now, which one has more DNA content? All of you. See, tell me, students, where do you find more thread? Students, where do you find more thread? You tell me, simple as that. Where do you find more thread? All of you. See, here we have less thread, here we have more thread. So here if I say the DNA content right now is 2C DNA content, and the number of chromosome, let's say, is 2N right now. Okay, number of chromosome right now is 2N right now because it's diploid in nature, and 2C is DNA content. This will now get converted to, students, what? Number of chromosome will remain the same. N basically means number of chromosome. Number of chromosome or set of number of chromosome. So diploid set. Number of chromosome is very same, but the DNA content is doubling right now. So it will become 4C. C is the DNA content there. C is the DNA content. So students, I want all of you to understand in DNA replication. See, first is cell growth. Second is DNA replication. In DNA replication, the DNA content is doubling here. DNA content is doubling. But is the chromosome number increasing? No. Chromosome number will remain the same because one centromere here, one centromere here. So number of chromosomes remains the same. DNA content will always double. Students, how many of you understood the concept? Students, how many of you understood the concept? All of you. Students, how many of you understood? Students, how many of you understood? All of you understood? Yes or no? So 2C to 4C the DNA content is becoming, but number of chromosomes will remain the same. Last is called as cell division, students. Last step is called as cell division. Students. Okay. Now that is actually cell cycle. Here is a quick definition for all of you students. Quick definition. Okay. The sequence of events. It is a sequence of events by which a cell duplicates. Cell is becoming from one cell into two cells. The cell duplicates its genome, the entire DNA content, synthesizes the other cell constituents. It means the other organelles. It could be endoplasmic reticulum, it could be your Golgi complex, it could be ribosomes, it could be centrioles. Everything duplicates and eventually divide into two daughter cells is called as cell cycle, students. It is called as cell cycle. Now if you ask me what are the phases of cell cycle, students, I'll teach you. Inside line blind I'll teach you. Parki I'll teach you. Paki I'll teach you inside line blind also today's class. Don't worry. Now what do I mean by phases of cell cycle, students? Phases of cell cycle. Now if I want to divide the entire cell cycle, I will broadly divide into two categories. Only two categories, students. Only two categories. Okay. Phase of cell cycle can be broadly classified into two separate categories. Okay. The first category is called as your interphase. The second phase is called as the M phase. Now let me ask all of you one simple thing. Every single examination, right? Every single examination right now, every single examination you are writing right now, do you prepare for the examination or do you simply go and write the examination? You prepare for the examination, right? All of you are right now preparing for examination. Yes or no? You are also called as NEET aspirants, right? You are aspiring, right? You are aspirants right now. So even a cell before actually dividing, it will prepare for the division. A cell cannot simply go and divide. Like how you cannot simply go and divide, right? You cannot just go and you just cannot go and write the exam. without even learning chemistry, zoology, botany, physics, you cannot write the go exam directly. You cannot go and write. Not possible. Similarly, students, similarly, when a cell has to divide into two cells, it has to prepare first of all. So if I say right now, all of you watching the lecture right now, who has not liked the video, quickly like the video, students. All of you right now are in interface right now. Students, all of you right now are in interface. Okay. So in interface, a cell basically prepares. It prepares. Okay. It is preparing. It is basically cooking. Okay. It is basically cooking here. It is cooking here right now. Now, if you ask me, sir, are there any phases here? Are there any division of your interface? Yes, it is. Yes, it is, students. For example, entire interface can divide into G1, also called as your gap 1 phase. Then we have S, called as synthesis phase. And then we have gap 2, also G2, called as gap 2 phase. So we have gap 1, S, gap 2. Now this is under interface. This right now is under interface right now, students. Interface right now. Okay. Now students, what about M phase? Now I will call M phase is also called as mitosis in this case. M phase is again further divided into two different phases. Okay. The first phase is called as chirokinesis, students. The first phase is called as chirokinesis. The second phase is called as cytokinesis. Okay. Chirokinesis as well as your cytokinesis, students. Is that clear to all of you? Now, students, can anyone tell me what do I mean by chiro? Anyone? Students, what do I mean by chiro? The meaning of chiro is nucleus. So in the chirokinesis, the nucleus is dividing, students. The nucleus is dividing. Okay. So in chirokinesis, the nucleus is dividing, students. The nucleus dividing. Now if I say chirokinesis can further be divided into four different phases called as PMAT. P-M-A-T. PMAT. Now what is the PMAT? What is the meaning of PMAT, students? Very simple. Okay. Right. The meaning of P here stands for prophase. All of the PMAT in the chart. Metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, telophase. PMAT in the chat, all of you. PMAT in the chat. Okay, that is your PMAT right now. That is your PMAT right now, students. Now, what about cytokinesis? Students, the meaning of cyto means cytoplasm. Now, once the nucleus has divided, the cytoplasm also needs to divide later on. The cytoplasm also needs to divide right now, students. Okay. Now, this entire PMAT, right? After PMAT, the cytokinesis again is divided into two categories. Cytokinesis is very different. Cytokinesis is very different in the case of your animal cell. It is different. It is different in the case of plant cell. It is two different things. So in the case of plant cell and animal cell, it is two separate things. Okay, students. So is that clear to all of you, students? Okay. Done, students? Done? Done, students, all of you? Any doubt till now? Students, any doubt till now? All of you, any doubt till now, students? If there is no doubt till now, I hope all of you are making notes also, students. I hope all of you are making notes. All of you making notes? Yes or no? All of you making notes? Yes or no, students? Because I want all of you to, you can make the notes, you can tag me, right? You can tag me also on Instagram if you want, right? So please make the notes also side by side. I'll give you the notes. Yes, I remember, obviously. Right, obviously I'll remember. Students have a good memory, right? Okay. Now students, I want all of you to understand this diagram right now. I want all of you to understand this diagram right now, student, this diagram right now. Now, can anyone tell me what is this entire blue region called as? Anyone? What is the entire blue region called as? This entire blue region. What do you call this entire blue region, students? All of you. Students, what do you call this entire blue region as? This entire blue region here. This entire blue region right now is called as interface. Okay, students, it is called as interface. All of you know that, students, right? Students, I will share you. Okay, motivating. Okay, I'll send today. Okay, now this entire thing is called as interface. Students, I told you interface is very long. Interface is very long. For example, you are right now preparing for the exam. You might be preparing for the examination for 1 year, 2 year, 3 year, 4 year, but examination is for only one day. Students, I told you here, all of you right now are in interface. All of you right now are in interface. You are preparing. You're cooking basically here. You're preparing right now, right? That is your interface right now. It is divided into G1, S, G2. So interface is divided into G1, S, G2. Can see here, G1, S, G2. Students, I hope all of you have this textbook, right? Students, all of you have textbook in your hand. Students, all of you have NCERT, apart from Aditya. Students, do all of you have NCERT with you? Yes or no? So I want all of you to open your book and write down in your book, textbook itself, mark this entire region as interface. Entire region as interface. Okay. Now, what about this entire black region, students? What do you call this entire black region? This entire black color region is called as what, students? M phase. It is called as M phase right now. M phase. Now, students, I want all of you to understand the M phase is further divided into this purple color part. You see this purple color part here. This purple color part is called as chirokinesis here. Chirokinesis. And what about the small pink color region? This small pink color region, students, that is called as cytokinesis, students. Cytokinesis. So, do all of you understand the cell cycle? This is the entire cell cycle. It is the entire cell cycle. Here we have the longest phase is called as interphase, G1S, G2. Then we have a short M phase, which is divided into chirokinesis, cytokinesis, and here we go. Did all of you understand, students? Did all of you understand? Yes or no? Students, yes or no? This entire thing is called as the phases of cell cycle, right here on your board. All of you. Understood? Students, all of you understood? If you understood, students, quickly add some fire emoji in the chat because I want to see some fire, students. I don't see any energy from all of you because this is quality content. Roasted pig, huh? Why are you roasting a pig? Why are you roasting a pig? Okay. Now, students, you might be wondering, sir, how long do you think the cell cycle actually takes place? Okay. Students, how long do you think the cell cycle actually happens? In the case of bacteria, first of all. Students, in the case of bacteria, how long do you think it happens? See, let me give you a small clue, students. It is somewhere between 10 and 30. 10 and 30. Okay. So, in the case of bacteria, the cell cycle will last for 20 minutes. That's all. 20 minutes, entire thing is done. Kel khatam. Full. Everything is done. Hello, pigs. Okay. Now, what about in the case of yeast? In the case of yeast, it takes around 90 minutes. Little more time. Now, what about, students, in the case of human cell? What about in the case of human cell, students? In the human cell, 30 hours, 2 days, 3 days, 4 days, 100 days? No. In the case of human cell, it is around 24 hours. It is around 24 hours a particular cell will divide. Now, I want all of you to understand this entire 24 hours is divided into two categories. 24 hours. 23 hours is one and 1 hour is separate here. Now, all of you tell me, students, as you know, already know, which phase do you think happens for the longest time? Do you think interphase is very long or do you think M phase is very long? All of you in the chat, quickly, students. All of you in the chat, tell me. In this entire 24 hours, do you think interface will be for long? Obviously, right? So this entire 24 hours, right? This 24 hours or 95% of time, and here we have 5% of time, right? This entire 23 hours, a cell will be in interface. Simply eat, sleep, repeat. Students, eat, sleep, repeat here. Eat, sleep, repeat, right, students, interface. But remaining 1 hour, it becomes hyperactive. It's like, today I will divide. Okay. So here it is the M phase here. So M phase is only for 1 hour. It is going to be for 1 hour. 95% of time and chilling, just chilling, students, chilling. Okay. So students, that is the reality. That is the reality, students. It happens in the case of our body. So in your body right now, this is actually happening, students. How amazing is that? Your many cells in your body right now are in interface, or many cells in your body right now are in M phase. Okay, are in M phase, right, students? So imagine this is actually happening in your body right now. Right now it is happening. Crazy, students. So all of you believe in yourself, right? This entire phenomena can happen in your body. It means crazy life you have. Okay. Now, students, here we have something called as G naught. I will come back to this later on. We will enter G naught. Okay, we will enter G naught later on. Okay, we'll enter G naught. Okay. So what is G naught? I'll teach you. Have some patience, Indu. Okay. Now I want all of you to understand here. Now what exactly happens in the case of G1? Okay. Now G1 phase is also called as first growth phase. It is also called as post-mitotic phase. Now you might be wondering why, sir? Why is it called as post-mitotic? Let me give you perspective, students. Let me give you a perspective. Listen to me very carefully. Listen to me very carefully. Okay. First we have what? G1, S, G2, G1, S, G2, M, G1, S, G2, M, G1, S, G2, G1, S, G2, M. Okay, so it is a cycle, right? This right here is a cycle. It is a cycle happening like this. It is a continuous cycle. It happens like this. It is a cycle right now, students. It is a cycle. Yes or no? Now, students, we are right now talking about G1. Students, tell me, can I tell G1 phase is a phase? It is a phase between M and S. All of you agree with me, students? All of you agree with me? G1 phase is a phase. It is a phase between M and S, and it is a phase which is happening after M. Yes or no? So can I call it post-mitotic phase? It is called as post-mitotic because it is happening after M. G1 is present, students. G1 is present after M. So I will call it as post-mitotic phase. Okay. So it is called as post-mitotic phase. It is actually the longest phase. It is actually the longest phase here. It is post-mitotic because it is happening after mitosis. Post-mitotic phase. It is a phase which is between. It is a phase which is between M and S. So it is a phase between the M and S. So between M and S we have G1. We have G1, students. Okay. Now I want all of you to understand, students. In this phase, what is happening? In this phase right now, the cell will continuously grow. The cell is metabolically active. Metabolically active and continuously grows. It is metabolically active and it is continuously growing. But there is no cell division. But no cell or no DNA duplication. Not at all. There is no DNA duplication at all. Okay. So students, I want all of you to understand, in the case of G1, longest phase, post mitotic phase, between M and S, cell is metabolically active and continuously grows. The cell is growing. The cytoplasm is growing here. But there is no DNA duplication. The DNA duplication does not happen at all here. Does not happen. So when do you think this DNA duplication, students, happens? DNA duplication happens in the next phase, that is called as the S phase or synthesis phase. The S phase or synthesis phase, students, in this phase what happens? That is DNA replication happens. So the DNA replication takes place in the case of S phase. This right here is a NEET PYQ, students. This right here is a NEET PYQ. When do you think the entire DNA duplicates? The entire DNA duplicates in the S phase. In the S phase, students. Okay, in the S phase. Now, students, apart from this, along with the DNA replication, there is one more organelle called as centrosome. Okay, centrosome. So centrosome also duplicates inside the cytoplasm. Inside the cytoplasm. If centrosome, students, if centrosome is duplicating, a centrosome has two centrioles, all of you know. One centrosome has two centrioles. All of you are aware of that, right? One centrosome has, right? One centrosome has two centrioles. One centrosome has two centrioles. So centrosome also duplicates in the cytoplasm. Then where do you think DNA replicates? Students, please remember, DNA replicates in the nucleus. So this entire thing happens inside the nucleus. It happens in the case of nucleus, DNA replication. Centrosome also duplicates, or one centrosome has two centrioles, it also duplicates. Now, but please remember, this is the most important thing where many students get it wrong. There is no change in chromosome number. Chromosome number will always remain the same. If there was a diploid number of chromosome, it will remain diploid. If it was haploid, it will remain haploid. If there are 46 chromosomes, it will remain as 46 chromosomes. The number of chromosomes will never change. It will never differ. Number of chromosomes will always remain the same. Okay. The only thing which changes, the only thing which is changing is the DNA replication or the DNA content. The DNA content, students, the DNA content will go from 2C to 4C. 2C to 4C, the DNA content is happening. Okay. It is happening, students. Okay. It will remain the same, students. It will remain the same. Okay. Is that clear to all of you, students? Yes. Students, I'm trying to write more. That's all. It will remain the same. Students, is that clear to all of you? Students, is that clear to all of you? Yes or no? Yes or no, students? Yes or no? This is what is happening in the S phase. Now, what exactly happens in the case of G2, students? G2 phase. Now, students, I want all of you to understand, students, it is the second gap phase. It is also called as pre-mitotic phase. Now, why do I call it as pre-mitotic, students? Now, all of you know, this right here is our G2. This right here is the M. Now, is it happening? G2 phase is happening before pre-mitotic. It is happening before M, that is G2 here. So, pre-mitotic phase is happening, students, here. Now, here, students, I want all of you to understand all the mitochondria and chloroplast duplicates completely, right? All of you should know there is something called as your tubulin protein. The entire tubulin protein also replicates. Tubulin protein also changes. All of you should know tubulin protein is part of your spindle fibers, right? It's part of spindle fibers that also completely replicates right now. Okay, so tubulin protein also replicates here, also replicates, right? Apart from the students, in general, there is an increase in protein synthesis. Protein synthesis. Now, why is this a general, students? Why is there a general increase in protein, students? There is an increase in protein synthesis because the cell has to now undergo mitosis. It has to undergo mitosis right now, because by this time the cell has to be completely prepared. Like, imagine, like, at the end of the examination, you do all the mock tests, you do a lot of mock paper, you do a lot of PYQs, you read the entire NCERT one more time, you go over all the scientist names, you go over the entire different types of your diagrams. So you are doing a final check right now. So this right here is a final check. So this is the final check here. So G2, let's go back, students. Here first we have G1 phase, longest, post-mitotic, between MNS, cell is metabolically active, continuously grows, but no DNA replication or DNA duplication here. Then we have the S phase, DNA replication happens here, centrosome also duplicates in the cytoplasm, there is no change in chromosome number. Then finally we have the entire G2 phase, pre-mitotic, it is happening before M phase here. Here mitochondria, chloroplast also duplicates, tubulin protein duplicates, and also in general there is an increase. In general, students, there is an increase in protein synthesis, students. Any doubt till now, students? Any doubt till now? Anyone? Students, any doubt till now? All of you? All of students? Any doubt till now? No doubt? Students, if you open your NCERT, here is your NCERT. Just download it. Okay. Now let's just go through NCERT. Let's see if you have covered every single point before we go to mitosis. Okay. Difference between G1 and G2. In G1 and G2, you'll see not much doubt, students. In G1 and G2, mostly the cell is growing cytoplasmically. The cell is growing in size and preparing. That's all. Mostly. Okay. Now, students, see this. Cell division is a very important process in living organisms. During the cell division of cycle, DNA replication, cell growth also takes place. I told you there is DNA replication, cell growth, all of it is happening. Okay. That's what answer. Where is it? Where did it go? The sequence event by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two rod cells is called a cell cycle. Although cell growth in terms of cytoplasmic increase, I told you, cell growth is increase in the cytoplasmic increase. Cell growth is increase in cytoplasm. Okay. Cell growth is increase in cytoplasm right now. Okay. Once again, students. See here. Increase in the cytoplasm increases here. Now, it's a continuous process. All of you told you, this entire G1 S G2, that the G1 and G2 happens here, cytoplasm increase. Although DNA synthesis occurs only during one specific stage in the cell cycle. Students, can anyone tell me where do you think the entire DNA synthesis happens? Anyone? The actual DNA synthesis happens in which phase? All of you students. All of you students. DNA synthesis happens in which stage? It happens in the DNA replication happens in the S phase. Okay. Now, students, the replicated chromosomes or the DNA are then distributed to the daughter cells by a complex series of events during the cell division. These events themselves under genetic control. Okay. Now, students, here. This is what we learned here. This is what we learned, students, today. Okay. Now, see, students, see this. A typical eukaryotic cell is illustrated by a human cell example. 24 hours, out of which 23 hours, right? Out of which 23 hours it is in interphase, 1 hour is proper M phase. Okay. East is 90 minutes. I told you all of you students. Okay. So here we have the interface and the M phase right now, students. See this. Okay. I told you the entire M phase is divided into chirokinesis and cytokinesis. Chirokinesis, division of your entire nucleus. Cytokinesis, division of the cytoplasm. Both of them. Chirokinesis is divided into PMAT. Cytokinesis is different in the case of animal cell and plant cell. Now students, here we have the different stages of interface. Different stages of interface right now. Interface, different stages right now. Okay. Now see what is given here, students. See what is given here. See, just read what is given here, students. Okay. The G1 phase corresponds to interval between mitosis and initiation of DNA replication. What do I mean by that, students? See here. G1 right now, if you go back here, I told you G1 is between M, G1, S. M is basically the mitotic, S is basically initiation of DNA replication. Initiation of DNA replication is happening here. Initiation of DNA replication is happening right here, students. Okay. That is what is happening here. Okay. See here. Between mitosis, the M, and initiation of your DNA replication. That is basically M, G1, S. Mitosis, initiation of DNA replication is your S phase here. Okay. Now during G1 phase, the cells are metabolically active and continuously grow, but does not replicate its DNA. Very important line. So in the case of G1, the DNA is not replicating at all. G1. Okay. S or synthesis phase marks the period during which DNA synthesis or replication takes place. So the entire DNA replication or the DNA duplication, it happens in which phase? It happens in the S phase. See here, happens in the S phase. Okay. So 2C to 4C DNA content is increasing. However, there is no, very important line, no increasing chromosome number. So chromosome number will never change. Chromosome number will always remain the same. Chromosome number will always remain the same, students. Okay. Now even after S phase, the number of chromosomes remains the same, that is diploid itself here. Now in animal cell, during the S phase, the DNA replicates. I told you, DNA replication begins in the nucleus. So entire DNA replication is happening in the nucleus, and the centrioles are replicating in the cytoplasm. Centrioles or centrosome. All of you should know, one centrosome has two centrioles. Duplicates in the cytoplasm. During the G2 phase, see, this much is only given about G2, student. This much is only given G2. In G2 phase, proteins are synthesized. So this is the main point here. The main point for G2 is that proteins are synthesized. That is the main point about G2. Okay, bye-bye. Okay, right. Proteins are synthesized in preparation for the mitosis while the cell growth continues. Okay, while cell growth continues, students. So with that part is over. Now we come to something called as G0. Okay, students. Okay, so please understand what exactly is G0 right now, students. Please understand what exactly is G0 right now. G0. Okay, see this, students. I told you, some cells in the adult animal do not appear to exhibit division. Some cells do not divide. I'll give you one simple example apart from what is given here. Okay, now one is called as heart cell. All of you should know, heart cell is a cell which does not divide. Now one more cell example I'll give you, students. One more cell example is your RBC. Students. Red blood cells. RBC cells also do not divide. RBC cells also do not divide at all. RBC cell is a cell which undergoes, obviously the birth is happening and it has its own graveyard. So once the life cycle of RBC is over, it will go and die somewhere. That is called graveyard, called as spleen. So in spleen, they go and die. They don't divide further, students. They don't divide. Okay. So heart cell and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or death. Students, all of you think, students, think in the chat, think. Do you think the cell cycle has to always happen continuously? Do we have, so let's say you are just sitting in your house right now and you're not injured, cells are very healthy. Do you think the cells have to keep on dividing? No. This entire cell division is a very energy requirement process. So if the cell keeps on dividing all the time, if a cell keeps on dividing all the time, there will be no energy left for you. So to make sure the cell cycle does not happen continuously, nerve cell also, yes, nerve cell is an example right here. Okay, some cells, right, they do not divide all the time. They will exit the cell cycle, and whenever there is a requirement, when let's say you have an injury, let's say my finger got cut, I have an injury in my finger. Let's say I got an injury. I was playing football and I got injured. Now to heal the wound right now, the new cells has to come here. The new skin cells has to form there. So in such case, mitosis will increase. Okay. So whenever there is a requirement, mitosis can happen. Okay. So see, students here. And many other cells divide only occasionally. Occasionally means rarely. Occasionally as needed to replace that have been lost because of injury or cell death. These cells do not divide further. Exit G1 phase and to enter an inactive stage is called as quiescent stage or G0 phase, students, here, all of you. So these cells called as your nerve cells as well as your heart cells, they do not finish the cell cycle. As a matter of fact, students, if you can notice here, right after G1, they will exit the cell cycle and they enter inactive stage. They enter a inactive stage called as quiescent stage. Okay, this right here is the inactive stage called as quiescent stage. It is entering right now. Quiescent stage, it is entering, it is chilling right now. Just chilling right now, students. Quiescent stage, just chilling. Okay, G0 phase, students, G0 phase. Okay, G0 phase. Let's just go back here. NCRT line, students, NCRT line. Okay, see, students. So do not further exit G1 phase to enter inactive stage called as quiescent stage or G0 phase of the cell cycle. Now cells in this stage remain metabolically active. The most important line of NCRT. The most important line of NCRT is that the cells are entering an inactive stage. The cells are entering an inactive stage called as quiescent stage or G0 stage. Are they metabolically active or inactive, students? In the chat, all of you. Are they metabolically active or inactive? All of you. All of you students. They are metabolically still active. So that is very important line which many students think that just because they're entering G0, they're not becoming inactive. They're not dividing, that's all. But they're metabolically active here. So they're metabolically active but no longer proliferate. That is, they no longer divide unless called upon to do so on the requirement of the organism. So based on the requirement of organism, they will divide, or else they will remain in quiescent stage. They will remain in quiescent stage. So all of you will remember, students, all of you remember that line. Very important line. Very, very important line, students. Okay. So students, I want all of you to understand. Can we just solve this part here? Let's just solve this part here, right, given here. Okay. You have studied about mitosis in onion root tip. It has 16 chromosomes in each cell. So every single cell has 16 chromosomes. 16 chromosomes. Okay. Now, can you tell how many chromosomes will the cell have at G1? Obviously, students, G1 it will have 16 chromosomes because chromosome number doesn't change. Chromosome number doesn't change. Yes or no? Okay. After S, it will still remain the same. In S, it will remain same itself because number of chromosome number never changes. DNA content changes, but number of chromosomes remains the same. Okay. And after M phase also it is going to be 16 itself because number of chromosomes does not change. DNA content is changing, but number of chromosomes remains the same. Okay, students. Okay. Now, what will be the DNA content of these cells at G1 as G2? DNA content will change, obviously. Let's say, students, let's say here in G1 the DNA content is, let's say at here, let's say we have your G1 S G2. This is your here chromosome here. So here we have 2 and 2C. In S phase the DNA content doubles here, so we become 2 and 4C. The DNA content is doubling. Now in G2 phase nothing happens, it remains the same, 2 and 4C itself. It will remain as 2 and 4C itself, students. It will remain as 2 and 4C itself. 2 and 4C itself, students. Okay, 2 and 4C itself will remain. Okay, 2 and 4C. The number of chromosomes is always the same. The DNA content doubles right here, that is in the S phase. Students, I hope all of you are able to understand the concept here. Very, very important concept, students. Okay. Now, in animals, the mitotic cell division is only seen in the case of diploid somatic cells. So the whole meaning, students, the whole meaning is that I will do reproduction this week itself. Okay. Now, what do I mean by this line here, students? Usually what happens, mitosis happens in diploid cells usually. In the case of animals, students, in the case of animals, a diploid cell undergoes mitosis. But in the case of certain plants, such as your algae, and even in the case of your drone, that is your honeybee, right? Here a haploid cell can also undergo mitosis. It can undergo mitosis. Okay, students, it can undergo mitosis. As you can see, students. However, there are a few exceptions to this, whereas haploid cells divide by mitosis. For example, male honeybee. Against this, the plant can show mitotic division in both haploid and diploid cells. So, in the case of plant cell, both a haploid cell and a diploid cell both can undergo mitosis. But in the case of animal cell, generally a diploid somatic cell will undergo replication. Students. Further, you will understand in next class, students. Students, that's it for today's class. Remaining chapter we'll do in the next class, where in the next class, students, I'll be taking your entire. In the next class, I'll be teaching you the entire, students, entire mitosis in the next class. Okay, the entire mitosis in the next class, students. Now, students, let's do some questions. Students, before I do some questions, here's a question for all of you. Okay. Because it is not dividing, because Saka, understand, in the case of your G2 also, is the chromosome dividing anywhere? No. So it will remain the same. It will remain the same, students. Okay. So 2 and 4C only will remain. So here is a question to all of you. Students, all of you quickly smash your like button, all of you. All of you quickly smash your like button. All of you. All of you quickly smash your like button. Here's the question on the board. Students, 1 second, let me just find something for all of you. 3n students, 3n very simple. No, students, 3n is your this thing. Remember, endosperm. Yes or no? 3n is endosperm, students. So the cell which doesn't enter your G0 phase is basically your meristematic cells, students. These are your meristematic cells which does never enter your G0. Never enter G0 are your meristematic cells. Which never enter G0 are the meristematic cells, students. Students, I want all of you to understand here. There are something called as checkpoints here. Now, what is a checkpoint? Now, whenever a cell is undergoing cell cycle, before it goes to the next stage, there is a checkpoint here. Unless the checkpoint is cleared, there will be no signal to go front. For example, students, you can see here, there is a first checkpoint is between right after G1. Right after G1, there is a checkpoint here. Okay, G1 checkpoint. It will check for nutrients, growth factors, DNA damage. If there is a DNA damage, students. So, because imagine if there is a DNA damage and it goes to the synthesis, the damaged DNA will replicate now. Yes or no? So this checkpoint will check for all of this, nutrients, growth factors, as well as DNA damage. Then students, right after G2, here, there's one more checkpoint here. Now this checkpoint will look for cell size, is it proper, DNA replication, if it has happened properly or not. If the DNA replication has not happened properly, it will send back again. It will send back again. So these two are the main checkpoints, G1 checkpoint and G2 checkpoint. Now apart from these two checkpoints, students, even in the M phase, there is one more checkpoint here. That is chromosome spindle attachment. This happens in the case of metaphase. It happens in the case of metaphase. This you do not worry, students. This I will teach you when I'm teaching you about your mitosis. But right now, remember, after G1 one checkpoint, after G2 one checkpoint is there. Two different checkpoints are there, students. Two different checkpoints are there. Okay. Let's see which questions you can solve actually. Students, I don't think so you'll be able to solve a lot of questions right now. Students, can you tell me what is the answer to this question? What is the answer to this question, all of you? All of you? Students, what is the answer? This is a NEET 2024 question. NEET 2024 question right here. Following are the stages of cell cycle. Okay. First we have gap 1. Then we have S, that is basically synthesis. 1, 2, 3. Then we have chirokinesis. Last we have cytokinesis. Last we have the cytokinesis. Yes or no, all of you students? Yes or no? Last we have the cytokinesis. Yes or no, students? So what is the answer? E, C. There's only one option. There's only one option, students. Students, remaining questions are generally, okay, solve this question, students. Solve this question quickly. All of you solve this question quickly, students. It's a fairly easy question. All of you, what is the answer? See, G1 phase, cell is metabolically active and grows. Okay. S phase, replication of DNA. Okay. G naught, cell is metabolically active but no longer proliferate, does not divide further. G2, synthesis of protein. It was a 24 question. So A is 4, there's only one option. There is only one option, students. Okay. Students, this is your homework question in the comments. Homework question. Comments. It's a 2023 PYQ. 2023 PYQ students, solve this question in the comments and you're dismissed, students. You're dismissed today. Students, again, I'm telling you, students, you need to see, do you realize in YouTube what happens is that classes can happen not in a very continuous fashion, right? I took human reproduction long back and still I'm not able to finish it because there's no proper slots for me. Yes or no? So if you're part of DNA batch, you already know. I have finished in DNA batch, I have finished cell cycle, I have finished cell the unit of life, I have finished entire living world, I have finished the biological classification. Now today I started the plant kingdom also. In plant kingdom today I was doing algae. Students, if you're not a part of DNA batch, students, again I'm telling you, there's a link in the description, you can go check. DNA batch has started. The prices are still low because after 1 week the prices are again going to go back to 6,000. Okay, so if you're not a part of DNA batch, students, please join the DNA batch because in DNA batch you will have all the notes, you will have all the live sessions properly planned throughout the year. By the end of December, the entire syllabus is going to be over, right? Entire syllabus is going to be over by the end of December, right? So if you're not a part of DNA batch, please join. Link is in the description. And students, sell the unit of life. I'll send the notes very soon. Okay, today only I'll send. Don't worry. In Telegram, that is Vedantu English Telegram, I will send the notes. All of you. Okay. So thank you so much, students. Bye bye. All of you. Take care and have a great day ahead. And I'll see you in the next class. All of you. Bye bye, students.